Hello, everyone. Welcome to today's webinar on how your donation will make a difference, this big give. So the big give will begin on Monday, the 3rd of December at midday, ending on midday, the 10th of December. So for 1 week only, you can double your donation at no extra cost, and this will go towards groundbreaking medical research projects. So we're delighted to welcome some of our research grantees today to talk about their projects and how your donation would really make a difference. So since 2017, through the Big Give, thanks to you, we have raised over £250,000 to invest into innovative medical research. And to give you just some examples of what a donation could do, a donation of £7.50 would be doubled to £15, which would buy sterile pipettes that researchers need to feed and maintain cell cultures. £50 would be doubled to £100, which could pay for 4 hours of medical research. £100 would be doubled to £200, which could buy an antibody needed for experiments to see how RP mutations affect protein levels within the light photoreceptors. So the Big Give is an excellent way that you can double your donation and double your impact to help us accelerate the search for treatments. So thank you to our panelists for coming along today, and please do feel free to put some questions in the question and answer box, as we'll have some time to go through that at the end. So I'm going to hand over to Kate now, who will be leading the discussion with our researchers.
I'm muted. Thanks very much, Amelia. And I'm really pleased to welcome 3 of our researchers today. So 2 senior researchers, Professor Lako from Newcastle University and Professor van der Spuy from UCL. And we're also joined by Elena, who is a PhD student at Oxford University, who has nearly completed her PhD. Elena, we'll start with you. If you could tell us a little bit about your project, please.
Yeah, hey, thank you for having me here. So yeah, this project so far has focused on comparing different gene editing technologies in targeting ABCA4, which is the causative gene in Stargardt disease, which I'm assuming many people are familiar with, and that is a macular degeneration. So specifically, there's tools that can change a single base pair, so like a tiny part of a gene, and that allows you to correct a specific mutation. And so I've been comparing 2 systems that target mutations in DNA or RNA to see which is more effective. And what we found so far is that it is extremely mutation dependent, but when it does work, the results are very encouraging. So for example, we've achieved high editing rates, and that's led to full-length protein, which is very exciting.
So can you just explain a little bit what you mean about it being mutation dependent, it not always necessarily working?
Yeah, so some mutations, for kind of unknown reasons, it seems like, I mean, I think it's like maybe biophysical or something, will affect how well these editors can like access the DNA or RNA, or just like access the target, and that will then affect the editing efficiencies. So some will get like 50% editing or something like that, and then others you get absolutely no editing. But like prediction tools aren't really good at like telling you where that's going to happen.
Okay, so I should actually perhaps just jump in and quickly explain that whilst the same gene might be involved in all these different cases of Stargardt disease, different people will have a different spelling mistake inside that gene that's causing their condition. So that's where you get, that's what we mean by when we talk about different mutations. It's the same gene.
Oh, sorry. Yes.
But yeah, no, it's fine. I just thought I'd just give people an idea of that, because I think it's quite easy for us to forget that we have all of these genes across inherited retinal dystrophies, and then within those genes there are different individuals will have a different part of the gene that's affected, and it's important for us to remember that as well.
Definitely. Yeah.
So tell us a little bit about our funding then, and how that's made a difference. Why is it important that charities fund PhDs like yours?
I mean, I think the main thing is that it like continues innovation and research in those fields, and especially like it gets people engaged in that research quite early on, and like fosters maintaining people in those fields, which I think in the long term like ultimately builds towards obviously like finding therapies and treatments and stuff. So I think it just, yeah, and in my case, it's been extremely helpful because it funded the whole 3 years and came with an amazing travel grant to go to conferences and, you know, talk to researchers from around the world also working in this field. And it kind of, yeah, it's good to know how tight-knit the community is and just like how willing people are to work together. So yeah. And
can you, yeah, go on.
I was just gonna say, like, before, like, like when I started doing this research, I didn't really know anything about the eye or about CRISPR or about Stargardt disease. And it really just kind of like captured my interest. And so then I really ended up applying for the PhD and got the funding and have since stayed in the field. And so that would just be like a personal example of that.
Yeah, because your previous studies weren't anything to do with the eye, where they remind me again, you did a undergraduate degree in
international relations and disease biology.
Okay, so quite different. And then I think you did a master's, did you?
Yeah, and that was in molecular bio, but my project focused on like using antibiotics produced by soil bacteria as like ecological pesticides. so
quite a big change. Yeah. So the fact that this PhD was available has brought you into this field, would you say?
Definitely. Yeah, absolutely. Yeah.
And can you tell us a little bit generally about how PhDs are funded and what the PhD funding landscape is like? Perhaps you've got some examples of like colleagues who've had a different funding mechanism, or
yeah, I think like the BRC grants are quite good, and like Wellcome Trust. Oops, is my internet gone? Oh no. And other like large scientific bodies definitely fund a lot of research. But I would say, like, in the grand scheme of things, IRDs aren't super well funded. But yeah, I think it's like, yeah, it's always important to keep funding the research and to make these grants available.
Thank you, Elena. I hope I completely lost connection there for a few seconds. So I hope that was okay. And what are your plans for the future? What are you going to do next off the back of having completed this PhD? I know you've still yet to finish things up, but have you got an idea of what you want to do next?
Yeah, there's a lot of really cool spin-out companies that work on like therapies for Stargardt specifically, or inherited retinal degenerations. So that would be very cool. Of course, like a postdoc would also be very interesting. But yeah, I think like a spin-out is exciting because it's kind of the next step after academia, in the sense of like, if scientific research like has led toward a treatment, then it does get spun out, and so it's like quite an exciting development in that.
So you're quite keen to watch these sort of scientific findings make their way forward towards therapies and towards our community ultimately in the end?
Yeah, definitely. Yeah.
Okay. It's great to hear that you want to sort of stay in this area. That's what we always hope when we fund a PhD. Obviously, somebody who's done a PhD can go off and do whatever they want to, but it's lovely to hear that you're keen to stay with inherited retinal, within the inherited retinal disease space. Thank you very much. I'll move to our two more senior researchers. Linda, can you first of all perhaps introduce yourself, just say a little bit about your research background and your interests?
Yeah, so thanks for inviting me. So I'm professor of stem cell sciences at Newcastle University. Well, my background, I did my PhD in human genetics and human development. So it was very natural that then I got very interested in stem cells. And so I went to do a postdoc at Durham University to work with mouse stem cells. And this was in a very topical time, because in 1998, as I was doing my postdoc, the first human embryonic stem cells were derived. And so that was a dream for me to actually go move my research from mouse to human. And so I applied for various fellowships and independent PI positions, and I got the one in Newcastle, which was absolutely brilliant, because there we had an IVF facility where I could work with the clinicians there to turn the ethically consented spare IVF embryos into stem cells. And again, there I made a lot of clinical connections, and it seemed like the connections with ophthalmologists was really the one that defined my path as a principal investigator. So first started working on the cornea, But then I realized that the cornea is really very nice, the front of the eye to study and do clinical trials is easily accessible. Retina was something that really stole my heart, I would say. It's such a fascinating tissue and all the connections, all the biology, all the networks. So I would say that now 80% of my research efforts and my team are actually in the retinal field using the stem cells.
And can you tell us a little bit about the projects that we're currently funding, that Retina UK is funding at the moment?
Absolutely. I mean, we've been very lucky. We have 2 concurrent projects funded by Retina UK at the moment. So I'll start with the first one, which is focusing on gene therapy to restore normal levels of a protein called PRPF31. So PRPF31 is part of a much larger machinery spliceosome, which is almost like a scissor that cuts out unwanted bits of DNA. So I can compare this with making a film. So when ones make a film, they would take many shots, but then to make sense of the story, you cut some of the shots and you connect them together. And that's what the spliceosome does. It just cuts the, let's say, non-coding parts of the DNA to make coding the ones that really give rise to the proteins. And we knew that mutations in some of the components of spliceosomes, such as PRPF31 or PRPF8, were giving rise to retinal disease only, despite being present in all the other cell types in the body. So again, this is almost like a puzzle that attracted us to ask the question, why? And we had a previous project that actually funded us by Retina UK. So we were able to find out that the retinal pigment epithelium and the photoreceptors, the light-sensitive cells of the eye, the rods and cones, were actually affected. And the way they were affected is that the expression of this gene and the protein was actually half of what it should have been. So the whole, this editing machinery was really stalling, not working, and actually miscutting other genes that are important for retinal function. But we didn't have any experience in gene therapy. We knew that we wanted to restore the levels. So luckily we got together with Professor Robin Ali. He was at UCL, now he's at King's College. And he joined us and his team. And so they've been able to produce those viral particles in which those PRPF31 protein is produced, but then it's easy to actually transfer into the eye. So far we've had really good results with the retinal pigment epithelium. We show that we can restore the physiological levels of this protein and can actually restore many of the functions. And in the last 6 months of the project, we're just moving on to see whether we can do the same with the photoreceptors. Then moving to the second project is slightly different. Again, we've always been very interested to produce photoreceptors that we can use for transplantation. So it's fine to do gene therapy in a retina where the photoreceptors are still there, so you can actually supplement the proteins that you need. But if the cells are completely gone, then you really need to replace them. And so we've been working with these retinal organoids, which are a lab mimic of the retina. And we can then generate photoreceptors there that are mature enough and form connection with the other cell types. So we can take those photoreceptors out, we can put them into mice that mimic the retinal disease, and somehow we restore partially the vision, which is very promising. However, if we're to do this in humans, we then will have to reprogram cells from every patient or person. And this is a long process, taking them back to stem cells, then making retinal organoids. And if someone needs a transplant very quickly, it's not possible because it will take a good part of a year to do that. Plus, it's also very expensive. So our approach has been to actually try and find what we call off-the-shelf, these pluripotent stem cell precursors. So all our cells have tags in the surface. So we know that if I have a cell from another person, my immune system will recognize those tags and will actually fight that. So we have actually created those pluripotent stem cells without those tags. So we've manipulated, either deleted or edited some, so to make them invisible to the immune system. And that has been the first year of the project. Has actually been quite intense and demanding because we're not performing one gene editing, we're performing 3 or 4 gene editing. But we've got there, and we are now moving to the next stage. So we want to make photoreceptors from those and then try and see if actually the human immune system really doesn't see them. So they evade that. And we call this hypoimmune or invisible photoreceptors. Again, very exciting project that hopefully one day will lead to a therapeutic product.
And it's lovely, actually, the way that there are links to other projects that Retin UK has either funded in the past or is funding now. So we funded Robin Alley quite extensively to develop a sort of pipeline of gene therapies and to really refine that gene therapy mechanism and those viral vectors. And then also we're also currently funding another researcher, James Sourden at UCL, who's looking at bioengineering a little scaffold for stem cells potentially, or cell-based therapies to sit in. So it's lovely how these projects potentially all dovetail together, either now or at some point in the future. So how do you envisage your current research progressing in future? Sort of what do you hope that these projects are springboards for?
Yeah. I mean, for the gene therapy project, we really hope that we can move to proof of concept clinical trials, especially with the FDA. Recently, they have approved products going ahead without being tested in retinal or organoid models without having to go to a mouse model or a primate model. So we would like, or our hopes is, or aims is to apply for a translational grant that will allow us to change this into a product for phase 1, phase 2 clinical trials, first to establish safety, then dosage. And if everything is successful, then it can be spread out. And because we're replacing the whole gene, it doesn't matter what mutation you have, provided that mutation results in reduction of the gene, then supplementation should work across the, across the patients that have got those mutations. In link to that, we actually also got funded by the MRC to look at the second protein, PRPF8. And that is slightly different in that it works in a negative way. So you have a protein that actually undermines the function of the wild type protein. So there we are really trying different approaches, but I think we've learned so much from the first one, the PRPF31, that we can now transfer the skills and the knowledge. For the second project, we really need to move to what's called good manufacturing practice. So if we want to have an off-the-shelf photoreceptors, we either need to repeat the procedures we've done the gene editing in a GMP cell line or adopt the cell line or create it to GMP conditions. And again, I mean, they are huge efforts. There's a lot of legislation and paperwork, but we are committed to that. And it could be a combination of either UKRI funding, or we're also looking at industrial funding maybe, because project of this scale is actually quite difficult to just only get funding from UKRI. You need a larger picture of that. But I think first and foremost, we need to finish what we have, but we do have those plans and ambitions for maybe about 2-3 years down the line.
Yeah, so I wondered if it was just worth jumping in and saying a little bit about, when you talk about UKRI, you're talking about public funders, you're talking about government-backed funding. Yeah. And just could we say a little bit about how perhaps the kind of projects that charities fund perhaps are quite important in providing a springboard for that? Because it'd be quite, am I right in saying it would be quite hard to go straight to UKRI?
Absolutely. I mean, without preliminary data, it's very difficult to go to UKRI. And for example, when we got funding for retinal organoid work about 12 years ago, was right at the beginning, and now is a really tool that everybody uses in the lab, but then was only a very few labs. And our first funding came from Retina UK, which allowed us to develop that really well. And then I got an ERC fellowship afterwards because I had enough data and I guess enough strength to get a much more prestigious grant, and then really rolled out the retinal organoid in my lab. So I think that preliminary data is very important, and the funding from the charity gives that, but also gives the continuity. UKRI is very, very competitive, and it's, you know, we've all been there. We've submitted grants and there has been shortage of funding, and they say the grant is good, but we have no funds. And then you lose skills in the lab because you lose those researchers that have really been inspired and done that work. And I think with the charity funding, I've always, such as Retina UK, I've always managed to keep sort of precious, very talented people in the lab to pass the knowledge on. But the other thing is also the high risk approaches. So whenever we've come to Retina UK, either retinal organoid or the hypoimmune have been quite high risk that they might work, but they might not work. Again, UKRI probably, they want a safe bet. They will not fund that. And luckily every time it has worked, but I think those high risk approaches are, you know, it's good that the Retina UK is funding them, because, and at every stage, you know, of development, whether it's early research or it's clinical, then we can make advancements that are really important for the patients. On top of that, I think the eye research is probably not on the forefront of the UKRI, or has not been. I don't know exactly the percentages, but when we compete to MRC UK, we always compete with other neuronal and mental health. So
yeah, I think it's a really tiny percentage.
It's a tiny, so
public funding that goes into medical research, I think, is something really small, like 1.5% or something. I don't know if Jackie, so, and when you think about the number of people affected by eye disease, it's really disproportionate.
But having this help has been tremendously helpful. And just to compare it, recently I've really become inspired of looking at retinal developments in other mental health diseases such as schizophrenia and autism. And this is a completely new field for me. And I've been looking at all the charities that fund their work as a way to try and approach them. And surprisingly, there isn't anything in this field like it is with eye research. So there are lots of charities, but they focused in making people's life easier, patient lives and the caregivers, which I think is fantastic, but it's actually very, very difficult. So in that respect, I think we're quite lucky. We have Retina UK or Retina Foundation Fighting Blindness or Macular Society Fight for Sight, that we can really tap into those resources.
Thank you very much, Linda. That's been a really good overview of the work that you're doing and how our funding is fitted in. Jackie, thank you for being so patient. Do you want to tell us a little bit about your background and how you?
No problem. So I actually come from a biophysics background, a little bit out there. So I actually did my PhD in biophysics lab that focused on structure function correlations in proteins, so did lots of protein folding assays and those kinds of things. I actually came to the UCL Institute of Ophthalmology through a Wellcome Fellowship, which was sponsored by Professor Mike Cheetham at the Institute of Ophthalmology. And essentially it was to apply some of my knowledge coming from the structure function background to explore a very new gene in the field, which had been shown to cause a very, very severe and early onset disease. So my background was in protein folding mechanisms, chaperones, etc. And all of a sudden we had something in the eye field, if you like, that, you know, where I could apply those principles that I'd learned. So I came to the Institute of Ophthalmology to work on this very, very newly identified disease-causing gene. And then I had a very linear progression over there, you know, moving from lecturer to associate professor, etc. But actually, I think that it's been very, very exciting. My interest has always been to understand how things work mechanistically, and then to take that understanding and develop into something more in terms of models and then ultimately, of course, therapeutics. And of course, it's been an incredibly exciting couple of decades to work in this field, because obviously, of course, first we had the advent of stem cell technology, which has really opened the door to be able to model retinal diseases, as Melinda has described, you know, with our retinal organoids. So I too have a very retinal organoid-based laboratory. We take iPSC and make retinal organoids and RPE. And then, of course, gene editing technology has also really revolutionized the field. So I think it's, you know, been an extremely exciting time to work in eye research. with the advent of these incredible technologies which we've been able to use to drive research forward. So the research in my lab obviously focuses on early onset, congenital and early onset inherited retinal degenerations, as well as juvenile childhood ones. And yes, I've been able to use these incredible tools to benefit the research that we're able to do in the lab. So that's my sort of history coming into eye research from a completely sort of unrelated background, but I'm really pleased that I have. I do think it is one of the best areas of research to be in, of course.
And nice to hear another familiar name in that story as well, because of course Mike Cheetham has funded by Retina UK in the past, and also has been a member of our medical advisory board. So you know, it's so nice to see everybody working together, and new researchers coming through from those more senior researchers as well. So can you tell us a little bit about your current Retina UK funding that you've got and the projects that relates to?
Yes, so I too am very, very lucky to be concurrently funded by Retina UK for 2 projects. One is a PhD studentship and the other is a project grant. So the PhD studentship is very interesting. I can see there's many parallels and overlaps, both with Elena, the work Elena has described, as well as Majlinda. I think this is both an advantage and disadvantage of going last. All the introductions have kind of been done. But the PhD studentship, the focus of the PhD studentship is to apply CRISPR-Cas9 gene editing also for a very specific and common mutation in the Stargardt's ABCA4 gene. And in this case, we are using a new technology called prime editing. So this project is in the very early stages, but there are numerous challenges to gene editing, including prime editing, one of which is efficiency, specificity, off targets, etc. So at the moment, what we do in the lab is we introduce the genetic variant of interest into an immortalized human cell line, in which we can optimize all the parameters of the machinery that we're using and the design of the machinery, both in silico and then to test and optimize that in vitro. So we've done that, and we can very efficiently edit this particular variant in our human cells. And then in the laboratory, very lucky, we have iPSC lines, so induced pluripotent stem cell lines that harbor the genetic variants that we're interested in, and we have isogenic controls for those. So genetically identical, except that they do not harbor the variation of interest. And we too differentiate those into retinal organoids. The ultimate goal of this particular project will, of course, be to see whether we can efficiently repair the genetic variant in the light-sensing photoreceptor cells in situ in our retinal organoid model. And there are numerous challenges to achieving that, as anyone in this field probably can testify. But yes, very, very exciting, and we've made tremendous progress in that area. And then the second project is a project grant. And for this particular project, we're going to the other extreme end, polar opposite end of the kind of the therapeutic approaches that you can apply for to inherited retinal degenerative diseases. So the first one I described as a gene-specific and mutation-specific therapy. And the second project we have is a completely gene-agnostic and mutation agnostic therapy. So this particular therapy is based on inherited retinal degenerations, many of which are characterized by the accumulation of a toxic molecule called cyclic GMP. It's a second messenger in the phototransduction cascade. So it's very important, plays a really important role in the transmission or the translation of a photon of light into an electrical impulse, which is then, of course, transmitted to the higher centers in the brain. And what happens in many IRDs is that there is a very toxic accumulation of the cyclic GMP molecule, which means that the channels that control the flux of ions across the membranes are constitutively open, and the cells effectively cannot respond to a photon of light, and they eventually die. And so in our lab, we again have retinal organoid models where we are modeling a very severe early onset congenital disease, which is characterized by this toxic accumulation of cyclic GMP. So for this particular project, we've partnered up with biotech, and they are in the business of developing cyclic GMP analogs aimed at rescuing this toxic accumulation, if you like. And so for the first time, we're going to be trying some of these cyclic GMP analogues in our retinal organoid model and using sort of state-of-the-art technologies to see if we can re-equilibrate the changes in iron homeostasis and calcium signaling that occur in these organoids as a consequence of this accumulation of the cyclic GMP. And so, yeah, this is an exciting project. I think, as I said before, it's, you know, it's gene agnostic, and there's a large number of IRDs which are characterized by this toxic accumulation. So the hope is that this will have broad applicability to a variety of different IRDs where they're underpinned by this accumulation.
I think it's nice that the projects we're talking about tonight, we've got the sort of crossover. We've got gene therapy and a very gene focused therapy, and we've also got these more gene agnostic approaches, the stem cell approach that Linda's looking at, and this approach to a sort of downstream problem that's happening with lots of different underlying genetic faults. And you know, it's really, really important to remember, and I'm really pleased that we're funding both, because it's so important to remember that so many of our community can't get a genetic diagnosis, or have a genetic diagnosis where nobody, you know, that it's not amenable to gene therapy, or nobody's working on that particular gene. I wondered if it was worth just quickly, we've talked a lot about retinal organoids this evening so far. I wondered if somebody would just like to quickly explain what a retinal organoid is.
Yes. Of course, yes, I should have done that actually. So,
okay.
So essentially, retinal organoids are derived from, as I mentioned before, induced pluripotent stem cells. And induced pluripotent stem cells are pluripotent, as the name implies, which means they are able to form any particular tissue in the adult human body. And the way we derive these induced pluripotent stem cells is we can obtain them from patients, and they can be, so we can obtain somatic cells from patients, to be that blood, hair, skin, urine, and those somatic cells can be reprogrammed into these induced pluripotent stem cells. We are then able to feed these induced pluripotent stem cells a mix or a soup of small molecules and signaling molecules in a very specific order, which will then induce these induced pluripotent stem cells to differentiate into our tissue of choice, which requires very good understanding of the developmental pathways which drive, you know, differentiation towards a specific lineage. So a retinal organoid is differentiated from an induced pluripotent stem cell. And what that means is if the induced pluripotent stem cell has been derived from a patient, the retinal organoid will be identical genetically to the patient from whom it was derived. So it's a very, very powerful tool to model diseases. And the retinal organoid itself, it basically mimics the development of the retina in utero. So it does take a very, very long time. It takes several months to make a mature retina, which is one of the challenges of doing this kind of research. It can take a very long time. And the retinal organoid is actually something that's extremely beautiful. So it's a laminated tissue. It has all the various layers of neurons that you would normally find in a neurosensory retina. So it's very, very beautiful. And that includes the light-sensing photoreceptor cells. So the photoreceptor cells are very special neurons that have a kind of an extended cilium, which houses the light-sensing material or machinery, if you like. And so the retinal organoids far exceed in superiority any other kind of model that we would have been able to use previously in vitro to model inherited retinal diseases. They're not perfect. They have their problems. Obviously, you know, they lack the underlying layer of cells, which is important for the homeostasis and maintenance of the retina, which is called the RPE. They lack, obviously, the vasculature and other things. However, they, as I said, they are far superior in terms of what we've had previously. And there are numerous groups worldwide that are working simultaneously on improving this model and ensuring that they can elaborate these proper outer segments, that they can maybe be co-cultured with the RPE, or both can be grown together, so that you have this homeostatic equilibrium occurring between those two tissue types. But yeah, I think they are very powerful, and they really have driven research in eye research forward in leaps and bounds.
Yeah, actually, and one of the newer projects we're funding that we've recently approved is actually looking at improving that model again even further. So yeah, that's nice to have as well. So thank you, Jackie, for just taking a little diversion there. In terms of going back to your current projects, if they're successful and if they, you have good outcomes from those, what would you envisage the next steps to be? I think particularly with the project grant, perhaps if what would the next step be towards getting a treatment that actually people could access?
So I think obviously the development of gene agnostic approaches for treatments of IRDs is extremely attractive and important, obviously, as they have the potential to treat a far wider range of genetic diseases. I think that if this works, obviously the next step would be to try to take this towards a translational pathway. As I said, we have partnered up with a biotech company who manufacture these. And so the idea would be to apply for much larger funding. Obviously, some of those cyclic GMP molecules are obviously licensed, patented, or whatever the case might be. So there are always, in the case of something where you're trying to take a product, into the clinic, there are those hurdles to overcome in terms of, licensing a product, etc.
Yeah.
We work very closely with the biotech company in order to do that. So the first step is obviously our proof of concept that this will work in this particular model. And if it does, then we need to obviously think about how we can take the next translational steps in terms of applying for larger funding body to larger amounts of funding, which will facilitate translation towards the clinic. There are so many steps in that pathway, and it takes, it can take many years, and it can be it can be very expensive as well. And so we need to make sure that our proof of concept preclinical studies are watertight before we take that step. However, we will be working together with collaborators in Germany and with the biotech industry. They, of course, want to apply this therapy to a wide range of IRDs as well. And this is the first time we've brought our IRD into the realm of being eligible maybe for this kind of gene agnostic treatment as well. Yeah.
You've mentioned proof of concept there, and I see proof of concept quite a lot in funding applications to us. And I wondered if you just wanted to comment a little bit, building on what Linda said about charity funding and where it fits in that pathway and that sort of proof of concept point, I guess.
I mean, obviously the charities fill a critical gap, as you've said before. I think that eye research in the UK amounts to the total amount of funding for research in the UK, only 1.3 to 1.5% is actually goes towards funding eye research. So the charities are critical in helping us to fill that gap, and I think Linda has pretty much said everything about the benefits of having charity funding in terms of advancing eye research. And I think we're in a lucky position to have charities which will specifically fund the kind of research that we're doing, and it is a very competitive environment, but it is still massively underfunded. And I think that we do need to we need to find a way for the charities and public sector to work together to perhaps find ways in which we can, support this kind of research further. In terms of the translational pipeline, I think it's very, very difficult because the amounts of money that are required, we normally go down the UKRI route in order to take that next step. There's various kinds of funding for that next step. So I think that charities are stepping up to the mark, though, with more bigger, larger translational kinds of funding coming on board to try to help researchers who do this kind of fundamental proof of concept research to take that step towards the translational pathway, it's complicated, it's difficult, especially if you're coming from a very fundamental background and you don't have experience in this, then actually taking that step towards the translational pathway can be very difficult. It's not only the massive amounts of funding that you require, but there's all the regulatory issues that require understanding and support. And so I think that charities have got a big role to play in facilitating and helping researchers to drive their research towards that translational pathway.
Yeah, I mean, it's something that we've definitely got an eye on, and I think we would like to be able to help researchers fill that gap. I think every medical research charity is probably really conscious that researchers, that gap in between, proving the concept in the lab and then moving to a point where you can start applying it in human trials is so difficult. And yeah, we would love to be able to fill that gap, but we can only do that with the support of our lovely supporters and growing our research strategy accordingly. Am I right, Jackie, in thinking that you had some funding from us several years ago?
Yes, I did.
Right?
I did actually. several years ago. And actually, I'm glad you raised that, though, because I think this is a perfect example of where charity support for fundamental research really can, spur on and support a whole translational pipeline. So the funding for that research was to have a look at essentially what we were the support that we had from Retinal UK was to have a look at new novel variants in a particular gene and to be able to confirm whether in fact they were disease causing or not. The gene we were looking at was very, very heterogeneous. In those days, we didn't have the masses of next generation sequencing data that we have now. And in silico tools weren't so good at predicting, whether you found a new genetic variation in a gene, whether that was actually causing the disease or whether that was just part of the normal spectrum of genetic variation, in the human genome. And so that funding was to help us to try to make those decisions with actual experiments in the lab. So in silico, in vitro functional assays, structure function assays, harking back a little bit to my background. So we could try to have a bit more than an in silico prediction as to whether a mutation was disease causing or not. And that's really, really important because the purpose of that project was to assist with the triage of patients for gene therapy, for a gene therapy approach. Obviously, you don't want to treat a patient with gene supplementation therapy unless you're 100% sure that the genetic variant they're harboring is, of course, the cause of that disease. So it was really important, first of all, to assist in the triage of patients. And also, I think Melinda mentioned as well, I'm in a very privileged position where I'm able to work with clinicians at Moorfields Eye Hospital. We have a very, very tight collaboration between the Institute of Ophthalmology and Moorfields. And so through that project, I was able to get access to clinicians and to patients with this particular disease. And we were able to get urine samples from these patients. They were pediatric, and then reprogram them into iPSC and to make retinal organoids. And in fact, we completed a proof of concept study of supplementation gene therapy in those retinal organoids, which very effectively rescued the phenotype. And in fact, we're just about to publish a first-in-man human study for that particular gene therapy. So it's all very exciting. So I think that's a really good example of how we've come all the way from fundamental research supported by Retinal UK to an actual treatment first-in-man study in the clinic.
That is a fantastic story. Thank you, Jackie. And just how long has that taken then?
That's such a brilliant question. Right, let me have a look at when I first got that funding. I think I have a date somewhere for that. So I think I was first funded by Retina UK for that. Must have been 2015 or so. So,
wow, yeah. Okay. So
yeah,
so 10 years nearly.
Yeah, 10 years nearly.
Yeah. Which actually, I mean, I know that well, you're sort of first in human testing. We know that Luxturna, which is the one gene therapy we have available in the NHS, I think that was something like 20 years from proof of concept in the canine model up to regulatory approval. So yeah, you're halfway to that. But it's fantastic that that's moved forward. Thank you, all of you, for those excellent overviews. We haven't got any open questions at the moment in the Q&A box. I'm just wondering, just out of interest, Elena or Amelia, have you thought of any questions that you'd like to ask Linda or Jackie during the course of them speaking at all? Elena, I just wondered if you...
Sorry, I didn't know who would go first. I actually didn't, but it was really, really, really interesting to listen to what you guys were talking about. It was, I don't know, it was very cool just to see like, yeah, the development of that. I guess like one question I have would be like, what was the process you went through to become a PI, and like, how did you go about that?
Jackie? For me, it was a traditional path. So I finished my PhD, did my postdoc. I applied for quite a lot of fellowships very early during my postdoc to Wellcome Trust and to the MRC. I got very close to getting them, but I didn't get them. And then I got the PI position at Newcastle. But once I got the PI position, those fellowships had really given me the experience of being independent. I was also quite lucky during the postdoc that I got freedom to operate. So during my postdoc, I applied with my PI for grants. We got both of the grants. And so I was, as a postdoc, I was running them. I was like a junior PI in his lab. And I'm actually very grateful to that mentor, because not many people give you that freedom and that experience. So fairly traditional path, not successful at fellowships, but eventually successful at establishing my own group.
Mine was a very, very linear progression as well. So as I mentioned before, I came to the Institute of Ophthalmology as a fellow, and I was sponsored by Mike Cheetham there. And similarly to Linda, I was very, very lucky. So I had the freedom, obviously, to conduct my own research as a fellow. After the fellowship, I stayed on to do a couple of postdocs, because I understood how competitive and how difficult it was to establish your own group. And also, I was thinking deeply about looking further afield as well. And there are obviously benefits to moving elsewhere. There are benefits to staying in the same place as well. There are pros and cons to both. But whilst I had those postdoc positions in the Institute of Ophthalmology, I too had the opportunity to write a lot of my own grants and have those grants funded. So I had freedom to operate, which was really, really important stepping stone when lectureship positions came open. And it really helped me to progress and to take that next step. So I think luck and timing, plays a role. It's great to be in the right time at the right place. Hard work, I guess, tenacity, perseverance. Many people try and fail and try until they succeed. It's very, very competitive and very difficult to progress up that career ladder. So yes, resilience and perseverance, tenacity, belief, obviously, in yourself that you're capable of doing what you set out to do. So I guess for many early career researchers, the doorway to an independent career path is through fellowship. So it's vitally important that PhD students explore the kinds of fellowship opportunities that are out there, or mostly postdocs will be exploring fellowship opportunities that are out there. Once you have a fellowship, it's very, very difficult as well, because you're not only normally supporting yourself and your own salary, you're running a group as well. And, if you want to move towards a tenure track path, then you need to have evidence that you can do that you can support yourself and support a group. So not only are you running a group, but you're madly applying for funding as well at the same time. So yes, difficult, but there's lots of support out there. The Institute, we have a lot of support. We have fellowship committees, we have mentors. There's a lot of support now in place to help early career researchers to take that step to become a PI in their own right.
Yeah, it's difficult, and I think we're really proud to be able to fund PhD studentships. And I think actually what we would like to do as well is start to fund early career researchers just slightly later in their career too. And I think that's something that actually we and the other sight loss charities, the research funding charities, would like to look at together. So again, something to keep an eye on. And I think perhaps just quickly I'll say before we wrap up, a little bit about how we decide what we're going to fund. Researchers apply to us for funding, and that application goes through a really rigorous process. Actually, both Linda and Jackie will, of course, been through that, and Eleanor's supervisor. So we have a medical advisory board. Initially, researchers apply to us with a preliminary application that's fairly short and just outlines what they intend to do. And even that gets scored by a medical advisory board who give up their time. They're all experts. They give up their time voluntarily to help us with our funding decisions. And then we shortlist and we invite researchers to make a full application, which is a really hefty document. I'm sure it takes you plenty of time to fill in those applications. And as you said, a lot of your time is writing these applications for funding. And they have to provide us with an awful lot of detail, and an awful lot of detail on the budget and exactly what they're going to do, and the background data. And that goes through external review. So we find people who could be from anywhere in the world who are experts in this particular field that the project relates to. They give us their opinion. And then our medical advisory board again reviews both the application itself and the external reviewers' comments and thoughts, and finally decides or recommends to our trustees which are the best projects and which ones should be funded. So it's certainly a rigorous and careful process, and we are very careful about how we spend the money that you've donated to us to enable us to fund research. But I think that just about brings us to the end. Amelia, did you have anything else you wanted to say before we close?
Yeah, I wanted to thank you all for coming along. And one question that popped into my head, as it seemed a sort of recurring theme, was on collaboration and how tight-knit the inherited sight loss research community is. So I know, Jackie, you were also mentioning about how you work with Moorfields. And I just wanted to ask what kind of collaboration you have with different institutions at the moment, because I think it paints a really good picture of the landscape, and you know, everyone working together means much faster progress. So I was just interested in that.
So I do collaborate quite widely with people at other UK institutions, in addition to, you know, Moorfields Hospital, as well as overseas. So I mentioned that I do have collaborators in Germany, collaborators in Paris, and I work with a number of people in the UK as well. So yes, it is quite a collaborative network. Obviously, it's very much dependent, you know, on the kind of work that you're doing. You quite often need to seek out new collaborations to help support your work, or to, you know, co-write a grant with, etc. I will say it has become a little bit more difficult to collaborate with people outside the UK for obvious reasons, but you know, it's just another sort of barrier that we need to overcome in order to work together for the best outcomes for our eye research. But yes, so I collaborate with people at Oxford University, at Montpellier in France, all over the place. I actually have quite a wide range of collaborators. Some of those are informal, and a handful of those are formal collaborations as well. I'm sure Melinda has lots of people that she collaborates with as well.
Yeah, absolutely. So as you said, Jackie, collaborate within UK, outside the UK. Some of my best collaborators are at Leeds and at Germany. And actually in the Retina UK funding, some of the funding has gone to our German co-investigator, who is wonderful at doing proteomics and all sorts of omics studies. What has been beneficial for me lately has been collaboration with bioinformaticians and AI. That is really is adding a new dimension, I think, to the data analysis as we compare those retinal organoids to the in situ retinas. We need, you know, those bioinformatics skills to show us if we're going in the right direction. But also with the AI mathematicians, we are at the beginning of some of those collaborations, trying to see if, you know, a lot of our work can be automated faster and much more objective. For example, an image analysis can take us a long time, but if we train the computer to do it, it can give us results much faster. And we're trying to implement that actually in the PRPF31 gene therapy project, so we can analyze many more of these images at shorter time. In parallel, we're also doing our manual imaging at the minute to validate it. But for us, those collaborations have really been new and telling us a lot about, you know, multi-dimensional data analysis that would have not achieved the traditional approaches.
Thank you for letting me know about that. It's very interesting. So just to wrap up, keep an eye out for when the Big Give opens on the 3rd of December, and double your donation, double your impact, help us support researchers like you guys, and keep a lookout for that, and help us to transform research and transform lives. And I'd also like to let you know to look out for the next webinar with Ask the Expert, to again open up important discussions on research. So thank you, everyone. Thank you for coming along and for your time, and keep an eye out for more information on the Big Give. And in advance, thank you all for your support. It makes a real difference in helping us move forward with research. So thank you.